Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine being able to see every single gene, every protein, not just on some flat slice, but inside a whole developing embryo, or say, a thick tumor, and seeing it all in glorious 3D with a nanometer precision, like really tiny details, and not just a few things, but hundreds simultaneously. Until, well, pretty recently, that was basically science fiction. But a groundbreaking new study in science has just made it real. It's nothing short of a paradigm shift. Welcome to the Deep Dive. Today, we're really digging into that revolutionary paper, deep tissue transcriptomics and subcellular imaging at high spatial resolution, and also looking at the analysis piece called Deep Nanoscopy, Mapping Genes in 3D Tissues. Our mission here is to explore how this new technique, it's called cycleHCR-ExM, how it was developed, what it can actually do, and why it's such a huge leap forward for seeing biology in its natural, messy, three-dimensional state.
Yeah, and to really get why this is so big, you need to understand the puzzle biologists have been trying to solve for ages. Think about it like this. For years, we could maybe identify where certain molecules were in a thin slice, like looking at a single frame of a movie. But what we desperately needed was to see the whole movie in 3D over time. Where exactly are these messenger RNAs and proteins inside a whole organ, especially in really dynamic things like developing embryos or complex tumors? We needed to link the molecules to the actual 3D structure. By the early 2020s, we had some pretty cool tools, spatial-omics tech like MERFISH or Slide-seq. They could map hundreds, maybe thousands of RNAs, which is impressive. But there was always a catch. They mostly worked on thin tissue sections, like really thin, getting deeper than maybe 150 micrometers. That was tough. Light scattering was a huge problem. It just blurred everything out. And the resolution was often diffraction limited, maybe 250 nanometers. Not bad, but
Not enough for the really fine details inside cells.
exactly. And then you had other methods. Some could go deeper into thicker tissues, sure, but you lost the resolution. You got the big picture, but missed the crucial details. And then there was expansion microscopy. Amazing idea, right? Make the sample physically bigger to see tiny things. It gave nanometer precision.
There's always a but.
Yeah, the catch was the processing. It was pretty harsh. Often it just, erased the RNA transcripts you were trying to look at in the first place, so you could see structure, but not the gene expression. It was frustrating.
So the core puzzle, the really tough nut to crack, was how do you get it all? How do you read hundreds of RNAs and proteins, too, inside thick samples like millimeters thick, and keep that subcellular nanometer detail? It sounds technical, but the biology behind it is huge. I mean, think about development, how an embryo forms, or how immune cells interact, or even how cancer spreads. These aren't flat 2D events. They happen in 3 dimensions. Try and understand them from slices is like trying to understand a whole city by just looking at one street map. You miss the connections, the depth.
Precisely. We needed a tool that could handle looking at many molecules, that's the multiplexing part, but do it deep within the tissue. This wasn't just about cooler pictures, it was about transforming fields like developmental biology, neuroscience, pathology, and even, providing a kind of ground truth for AI, looking at histology slides, making sure the algorithms are seeing what's really there. So yeah, that huge gap in technology was the real driver for this research. There just wasn't anything that could profile, say, dozens or hundreds of RNAs and proteins together in tissues thicker than 0.5 millimeter and keep that spatial resolution around 100 nanometers. Current methods? They either missed the long-range stuff because they were stuck with thin sections, or they caused you to lose molecules if you tried to clear thicker tissue. It was a trade-off nobody wanted.
Okay, so faced with that challenge, what was their big idea? How did they plan to bridge that gap?
Their core hypothesis was pretty bold. They thought, what if we could combine 2 powerful ideas? What if we married isotropic tissue expansion? That's the making it bigger part, but doing it evenly in all directions.
Like blowing up a balloon perfectly evenly.
Exactly. Combine that with a very gentle, cyclic form of a technique called hybridization chain reaction, or HCR. HCR is a way to label specific molecules. They figured that doing this gently and in cycles within an expanded tissue could finally beat the light scattering problem and stop the molecules from washing away. That's the heart of what they call true deep nanoscopy.
Okay, so combining expansion and the cyclic HCR, how did they actually make that work? What's the clever part in the design of this cycleHCR-ExM?
Well, the really key innovation, I think, was coupling a redesigned set of HCR probes, the little tags that find the RNA or protein, to a special kind of hydrogel. This hydrogel swells up 4 times its original size. And crucially, as the tissue embedded in it expands, the gel physically traps the nucleic acids and proteins. It locks them in place.
Ah, so the expansion itself doesn't wash things away because the gel holds on to them, like setting them in Jell-O before you make it bigger.
Kind of, yeah. That's a good way to put it. The gel matrix preserves everything while making it optically clearer and physically larger, which solves the scattering and resolution problems.
And you mentioned redesigned probes. What was special about them? How did they improve the signal?
Right, this is super clever. They engineered something called split initiator probes. Think of them as having a 2-part safety mechanism. They only really start the signal amplification when both parts bind correctly to the target molecule. This drastically reduces off-target binding, meaning much, much less background noise. You get a cleaner signal.
Okay, less noise. But how do they make the signal strong enough to see deep inside?
That's the other part. The amplification uses orthogonal hairpin pairs. These are like little molecular building blocks that carry fluorescent dyes. Once the initiator probe binds, these hairpins snap together in a chain reaction, building a long, bright polymer right there at the target site, embedding the fluorescent signal directly into that expanded hydrogel matrix. Orthogonal means they design different sets that don't interfere with each other.
Wow, okay. That sounds complex. How did they even test and validate something like this on thick tissues? It sounds like it would take forever manually.
It would. And that's where the automation comes in. This wasn't just a benchtop chemistry trick. They built a system. They engineered a pretty sophisticated robotic microfluidic stage. This robot handles the whole cyclic process, adding probes, washing, stripping them off, adding the next set. It can run 20 cycles automatically on samples up to 800 micrometers thick, like whole mouse embryos.
20 cycles?
Yeah.
How long does each cycle take with the robot?
Get this, less than 2 hours per cycle. Manually, it used to take something like 8 hours. So the robot makes these complex multi-target experiments feasible.
Okay, so automation handles the labeling. What about actually seeing the signal? What kind of microscope do you need?
For imaging, they used lattice light sheet microscopy. It's a very advanced technique that shines a thin sheet of light through the sample, minimizing photo damage and getting really clear images quickly. With this setup, they achieved an effective resolution of about 70 nanometers laterally, and they could detect single molecules for, amazingly, 254 different genes plus 8 different proteins in the same sample.
Whoa, 254 genes plus proteins. How do you tell all those signals apart? Doesn't it just become a mess of colors?
That's where those orthogonal barcodes come in handy again. They cleverly designed the labeling so they could read out 12 different genes in each imaging cycle, using distinct fluorescent colors and barcode combinations. It avoids spectral crowding, too many signals overlapping.
So it's not just one innovation. It's the chemistry, the probes, the hydrogel, the robotics, the microscopy, the barcodes, all working together.
Exactly. And it wasn't like they got it right on the first try. It was a real iterative process. Design, build, test, analyze, optimize. They modeled how the probes would behave versus how much the tissue expanded, tweaking probe lengths, buffer chemistry, all that stuff. That optimization process actually boosted the signal-to-noise ratio by 5 times compared to their early attempts. Really meticulous work.
Okay, so they built this incredible machine. What did they actually see with it? What new biology has it uncovered in 3D?
Well, one of the showcase experiments was looking at whole mouse embryos, very early in development, around day 6.5 to 7.0. They mapped those 254 genes, which are markers for different cell lineages, across the entire embryo, getting data down to about 310 micrometers deep. Then, by analyzing all that gene expression data from individual cells, they could computationally identify 9 distinct cell populations. And these populations matched up perfectly with known developmental lineages. But now, they could see exactly where each lineage was located in 3D space within the intact embryo.
So it's like a detailed 3D map of cell types during early development.
Precisely. But it gets even cooler. Because they had single-cell resolution and the 3D context, they could look at gradients of gene expression, how gene activity changes across space. And what they found was fascinating. Previously unseen gradients of RNA molecules running from the top to the bottom, dorsiventral and front to back, anterior, posterior of the embryo.
Unseen before. Why? Because people were only looking at flat slices.
Pretty much. Existing models of how these developmental signals, these morphogens, spread were based largely on 2D data. This new 3D view suggests it's more complicated. It strongly hints that the positional information telling cells what to become is organized in complex three-dimensional lobular niches, not just simple, flat gradients. It's like discovering the landscape is actually mountains and valleys, not just a flat plain. It could rewrite parts of developmental biology textbooks.
Wow, that alone sounds transformative.
Yeah.
But you mentioned it does proteins, too, and subcellular structures.
Yep, it's versatile. They showed that by combining it with the expansion, they could visualize 10 different structures inside individual cells, things like the nucleus, mitochondria, cytoskeleton, and mouse embryonic fibroblasts. Because of the expansion, they saw these structures with incredible detail, revealing really intricate architectures within the cell.
So you get the gene expression map and the detailed cellular context.
Exactly. And they even demonstrated unified RNA and protein imaging in the same sample. They did this in slices of the mouse hippocampus, a key brain region. There, they could see complex gene expression patterns alongside variations in the structure of the cell nucleus, specific to different cell types. It starts to let you link gene activity directly to cell structure and function in a really integrated way within the tissue's native 3D environment.
Okay, this really does sound like more than just an improvement. Why are people calling it a paradigm shift? What makes it qualitatively different?
It's because it hits this sweet spot that nothing else could. It simultaneously pushes the boundaries on multiple fronts. Depth, going deep into thick tissue, scale, hundreds of molecular targets, and resolution, nanometer-level detail. Before this, you had to choose. You could get maybe 2 out of 3, but not all 3 together in one package. This technique defines a new Pareto frontier, basically meaning it's the best across all these critical parameters at once.
So it outperforms existing methods like SEMFISH, MERFISH, ExSEC, but in different ways for each.
Right. It goes deeper than standard SEMFI-hash, has better resolution than MERFISH often achieves in practice, and handles way more targets than early versions of ExSEC. It kind of cherry-picks the best features and combines them, and the motivation was clear. We have these huge projects building cell atlases, mapping all the cell types in an organism based on their molecules. But without tools like this, those atlases risk being just, well, two-dimensional cartoons of 3D biology, as the researchers put it. This makes true 3D atlases a reality.
It sounds like a massive undertaking. You mentioned automation, advanced microscopy. This couldn't have been done in just one lab, right?
Absolutely not. It was a major collaboration across 3 big research centers. You had HHMI Genelia Research Campus, focusing on the chemistry and building that crucial automated fluidic system. Then the Broad Institute's imaging platform brought in the lattice light sheet microscope expertise and, importantly, the computational power to handle the enormous data sets. We're talking 4 terabytes of data per sample. They needed a GPU cluster just to process it. And UCSF's Sandler Developmental Biology Lab provided the essential biological samples, those carefully staged mouse embryos, and the expertise on which genes to actually look for, the lineage markers.
So a real synergy between chemists, engineers, physicists, computer scientists, and biologists.
Exactly. And academia only, multi-site collaboration, working in this agile, interconnected way. It's a great model for how big science can get done.
Can you highlight some of the key people whose specific contributions made this possible? It sounds like diverse expertise was critical.
Definitely. You have to mention Alina Petrovica, the lead author. She's an organic chemist who became a microscopist. She was the one who really conceived that crucial redesign of the HCR probes, the split initiator idea. Apparently, she showed real high-risk tolerance in ditching older designs that weren't working well enough.
So fundamental chemistry insight was key.
Absolutely. Then there's Carlos Bechtold, the engineer who actually built the Arduino-controlled microfluidic system. Taking that cycle time from 8 hours down to 2, that made the whole approach practical, engineering brilliance. And Ling Li, the developmental biologist, played a vital role in standardizing how the embryos were prepared, cleared, and mounted. That ensures you can actually compare results across different experiments, which is crucial for biological studies.
And the senior figures.
Well, Eric Betzig, one of the senior PIs and a Nobel laureate for microscopy, provided that strategic vision, pushing to combine the lattice light sheet, his own stomping ground, with expansion microscopy. It was a bet on a complex, cross-disciplinary approach, not just tweaking existing methods. That kind of leadership helps pull these big projects together.
Okay, so placing this work in the bigger scientific landscape, what's its impact on theory, technology, and application?
Theoretically, as we discussed, revealing those 3D RNA gradients really challenges and refines our models of development, particularly how positional information works. It pushes developmental biology theory forward. Technologically, it just sets a new standard. High multiplexing, nanometer resolution, millimeter depth, all in one. And crucially, they've open-sourced the protocols and designs. That's huge for accelerating adoption by the wider research community. No black boxes.
In applications, where could this go next?
The potential is massive. Imagine using this to see exactly how deep a cancer drug penetrates into a solid tumor and which cells respond, or mapping the connections between neurons in the brain at the synapse level while also seeing which genes are active in those connected cells. Whole brain transcriptomics with synaptic resolution. That's a holy grail.
It sounds almost perfect. Are there any limitations right now, things it can't quite do yet?
Yeah, there are a couple of things to keep in mind. The current chemistry uses a stripping step at 37 degrees Celsius to remove probes between cycles. That temperature might damage some very sensitive proteins, what they call heat labile epitopes, so it might not work for every single protein target out there. And the other thing is the equipment. Because the sample expands 4 times, you need special microscope objectives, ones with a high numerical aperture for resolution, but also a long working distance to reach deep into the expanded sample. Not every lab has those specialized objectives readily available, so that could be a barrier to entry for some.
Okay, so some technical constraints still, but the overall picture is pretty stunning. It really does feel like cycleHCR-ExM crosses a major boundary. It's enabling this genuine 3D multi-omic nanoscopy that people have dreamed about, a true game changer for developmental biology, cancer research, neuroscience, probably fields we haven't even thought of yet.
Absolutely. The implications for just fundamental understanding are profound. How embryos form, how tissues are organized, how diseases mess things up. We can now see these processes playing out in their full 3D complexity right down to the molecular level. It's like getting a completely new pair of glasses to look at the biological world.
So it leaves us thinking, what previously unanswerable questions in biology might you, our listeners, explore now that this kind of tool exists? Now that we can map genes and proteins in 3D tissues with this incredible precision, how could this fundamentally shift our view of life's complexity?